available to the public answers to many of the questions that have circulated. As you may have read, about 45% of the US population who has not got the vaccine yet does not intend to get the vaccine because they have doubts on the vaccine. And I think as scientists, it's very important to take any doubt seriously. And so today we're also here to ask our experts to discuss scientific basis for doubts. It doesn't matter. No question is a stupid question. So I want to start. Usually you thank people at the end. I think I want to start, use this time to start with thanking actually the people who really made it possible to organize this meeting. Of course, the volunteers from Professionisti Italiani at Boston, Francesca and the others and Alessandro, who spent quite some time planning the content and finding a way to make this happen. I want to thank, on behalf of all our volunteers, also the other groups that support this event, the other group of Italian professionals in Philadelphia and in DC, in Washington, DC. And also the 3 committees groups who are supporting the event, the committees in Boston, Miami, and San Francisco. Committees is a committee for Italians abroad, and they really serve a function of connecting the community also to our institutions and bringing the community's problems and requests, liaising for them with the institution. So I think their participation is a very important sign of their activity. And today we also have the support of the Comitato Generale degli Italiani all'Estero, which is a more international and global community. And I want to thank Silvana Mangione and Vincenzo Arcobelli for helping with that participation and support as well. And of course, last but not least, a big thanks to our Consul General and Vice Consul, who very open-heartedly and immediately supported the proposal. We had a similar discussion, a similar panel last year with the same 2 speakers, whom I thank very much for giving again time to us for this. And with the Consul, we immediately had aligned on the need to continue this discussion with the public. I want to leave now to the Consul room for introductions.
Thank you, Giovanni, and good evening, everyone. As Giovanni was saying, I think the theme of the event is what brought everyone together. The many organizations, of course, Professionisti Italiani at Boston and in other cities, Comites, CGA, as well as ourselves, of course, because it's essential for the public to be able to ask relevant questions to scientists. I think that we're all used to all the information that is in social media and so forth, but we're all aware of all the myths and mistakes, false beliefs that surround COVID and vaccines specifically. So I think that is essential for the information to be shared by the people who know what they're speaking about, such as the 2 very, very important speakers that you will have today and that Giovanni will introduce in a few minutes. So it's my pleasure to be here tonight. I think it's very important that we all listen and that we all ask questions so that we can go back into our communities and share the information. So without any further ado, thank you very much, Giovanni. It's very exciting to be here, and it's, as always, very exciting to be with distinguished speakers of the Boston community, of the Italian Boston community. So thank you very much, Giovanni, to you and your volunteers for taking the lead and organizing this event. And I give you the floor so that you can introduce the speakers and we can start. Thank you.
Thank you, Federica, and double thanks to you for attending this event from Italy, where it's pretty late at night. It's very important for us to have your support and presence continuously. So of course, we want to leave the floor to Professor Vespignani and Dr. Carfi. Maybe I'll shorten their profile introduction because both of them have a long and outstanding career. It will take longer to describe their career than for them to give the talk. Professor Vespignani is Sternberg Family Distinguished Professor at Northeastern University, where he also founded the Northeastern Network Science Institute. He gained a PhD and training in physics in Rome. Then he moved for his postdoc at Yale University and then Leiden University. He has worked at a number of prestigious centers, such as the Center for Theoretical Physics in Trieste, University of Paris, and National Council for Scientific Research. He then became professor at the Indiana University and fellow at the American Physical Society, member of the Academy of Europe, and fellow of the Institute for Quantitative Social Sciences at Harvard University. Professor Vespignani focused recently his activity on data-driven computational modeling of epidemic and spreading phenomena and the study of biological, social, and technological networks, which is clearly one of key reasons why we invited him to our panel. Dr. Andrea Scarfi is currently head of research at Moderna, where he has been since 2017. Prior to that, Dr. Scarfi has worked at Novartis Vaccines and at Angeletti in Italy before coming here. He was a postdoc at Harvard University and the Children's Hospital. Prior to that, he was at the Grenoble University, and he obtained a degree in physical chemistry in Pavia and a Master of Science in Biochemistry in the UK before moving abroad again. So I'll leave it to, I think Alessandro was going to speak first, correct?
Yep, I think yes.
Thank you again, both for your double participation to our discussions in November and today.
Thanks a lot. So it's a pleasure to be, to have this chance to try to make some summary or analysis of what is the situation now. And I'm as you know, you were saying, it's important that as scientists that we try to communicate as much as we can and answer questions. This is we are still at a moment in which there is really a need from the public perspective of understanding and try to have a better grip on what could happen in the next few months. Unfortunately, although we are in this pandemic since almost 2 years at this point, I think we are still a little to go before we can say we are out of it. And I think especially in the last couple of weeks, a few dark clouds have started to appear at the horizon, and some I start to receive, for instance, some questions of people who is worried about, and so I think it is a good moment. There have been a lot of excitement because of the vaccine, and we have seen these dramatic improvement across all places in the past months. So now the situation is slightly changing, and I think it's important to try to understand what is going on. And I think a good point to start is well, the fact that the pandemic now is I would say is has two different velocity. One velocity is in countries that have a large had the chance to immunize a large fraction of their population, and there is another velocity, another completely different trajectory in countries that unfortunately were not able to vaccinate important fraction of the population. So that's I think is the first the first data point we have to reason. And so at the same time, even in countries which where the situation was improving quickly and the vaccine seemed really to put a hand to the major crisis. You know, we have seen recently in the last few weeks some sign of growth of again of number of cases, and somehow the epidemic activity is picking up. Unfortunately, this is due to the fact that we are experiencing something that most of the agencies and the epidemiologists working on the field were saying even a couple of months ago. So a new variant of concern, the Delta variant that was originated in India several months ago, started to spread across multiple countries. This is a variant was increased transmissibility, and this is a major increase of transmissibility, if you want, because it's actually it's more transmissible than the B117 variant that already created some issues during the past spring. So we are talking about a variant that has almost twice as much the transmissibility of the original SARS-CoV-2 wild type. And this is what has generated is the activity, the epidemic activity that in many European countries started to manifest a few weeks ago. Actually, the UK were the first country to single out this major problem. They started to see an increase of activity and infections, especially in the non-vaccinated population, in the younger age bracket. And the fact that since in all countries we were relaxing the mitigation policies, the non-pharmaceuticals intervention, the social distancing, the use of masks, etc., this variant is now generating some concern. In the US also, in the last 2 weeks, we have seen the epidemic activity picking up with increase in terms of number of cases and hospitalizations, and this is this is a tale of two countries as well within the United States, in the sense that some states have reached a very large number of vaccinated people. They were able to have very vigorous vaccination campaigns. Some other states are lagging behind, and in those states, the variant, the Delta variant, is has a more fertile ground to gain in terms of transmission and number of cases. This again stresses infinitely the importance of vaccination. So the fact that even in a country like United States, where there is abundance of vaccine doses, the hesitancy of people in some states is generating a situation that might be complicated and might give rise to surges in not just in the number of cases, but also in the number of hospitalization and eventually deaths. I think it's Andrea will talk about the fact that the vaccine is providing an extraordinary protection, and is especially these variants are notable after 2 doses to how to say, to evade the vaccine protection, especially for what concern the protection for severe diseases, and so hospitalization and deaths. But I just want to close here with my 5 minutes, just saying that unfortunately, what we have seen is that those variants have emerged in places with large transmission and large number of cases. We had the P1 in Brazil, we had the B135 in South Africa, we had the B117 in the UK, the Delta in India. So these are all places where there was a lot of transmission, a lot of how to say, of chances for the virus to mutate, and this is why we have to think about this pandemic as a global event, and not just think that we can put an end to the pandemic within the border of a single country. We really need to devise strategies to distribute the vaccine to other countries that can't afford in terms of logistic, as well as acquisition and distribution of the vaccine. And this is in a sense, as I always say, is the donation and taking care of other countries is on one side the most generous things that we can do, and at the same time the most selfish, in the sense that is also the best way to protect ourselves from other possible how to say, mutation of the virus and dangerous threats. So I think I can stop here, and I want to give the floor to Andrea, and then I'm happy to take all the questions that you might have.
Okay. So Giovanni, do you want me to go ahead? Okay.
Yeah, no,
So
yes, thank you. Go ahead, Andrea.
Okay, thanks. I'll share a couple of slides so to make the discussion simpler. If I can find it, the screen.
È il pulsante in basso, share screen.
Yeah, yes, right. Thanks.
Yeah, it's easier to develop a vaccine for COVID than to share on
Absolutely.
Zoom for you, Andrea.
Yeah. Okay. So I think you can. Hopefully, you can see the slides. And
Yes.
Yes.
Okay. So I will. Sorry, it's just my computer is a little bit slow. So. Yeah, I'm changing the presentation. Okay. So I would like just to remind you a little bit about the vaccines and their efficacy, and then I'll touch up on the variants, just to facilitate the discussion later. But I would like to remind you that the one aspect of that is often also brought up from people that are have concerns about the use of the vaccines is the speed at which the vaccines were actually developed. And this slide really depicts some of the key steps that were followed for the development of the Moderna vaccine against the SARS-CoV-2, but the Pfizer vaccine was equally fast. And some of the key features were that we were able to really develop the initial vaccine in 63 days, going from the sequence of the virus to the beginning of the phase 1 study on March 16. And by the summer, we were able to start a large phase 3 clinical study to look at the efficacy of the vaccine in prevention of COVID-19 disease. And by the end of the year, November 30, we had the initial phase 3 data that showed the vaccine was highly efficacious, and that allowed for emergency use authorization here in the US, and then for a conditional marketing authorization in Europe in January. Since then, the vaccine has been authorized in a number of different countries. But during the development of the vaccine, we did both non-clinical studies, so in animals, to show the immunogenicity, protection, and safety, and in parallel studies in humans. And all the different phases of development were followed. We did a phase 1 study, which is just a safety study, then followed by a phase 2 that is more, exposing a larger number of subjects to the vaccine, and then the efficacy study. And what is important to emphasize is that two reasons allowed us to develop the vaccine very quickly. One is the technology. In our case, the use of the mRNA technology allows very fast development, because we don't need to grow a virus or cells, but it's an in vitro process. But then equally important, or if you wish even more important, is that we had both collaboration from regulatory agencies that worked very closely with all the pharmaceutical companies in developing the vaccines. And I think it was also the financial support by the US government through the Operation Warp Speed that allowed us to take financial risk in the development of the vaccine. So, but no risks were taken, or additional risks were taken for the safety of people involved in the clinical studies. And to summarize, the phase 3 study was a 30,000 subject study. 50% of the subjects received 2 doses of the vaccine, 100 microgram dose, and 50% received just a placebo, a saline solution. And what we were able to show is that at the phase the interim analysis, so when you initially look at the efficacy of the vaccine, of the 196 cases that were documented, 185 were in the placebo group, and only 18 cases were in the vaccinated group, showing that the vaccine had a very high efficacy, above 90%, so 94.1%. So this was a prevention of a symptomatic disease, but was also critical to look at prevention of severe disease and hospitalization. And again, at the same time point, we saw that there were 30 cases in the placebo group and none in the vaccinated, for an efficacy of 100%. So the vaccine clearly provides a strong protection from severe disease, prevents hospitalization from the virus that was circulating at that time. Since then, we have done a number of other studies in adolescents, 12 to 17 years old. We are conducting studies in the immunocompromised, and also study now in the maternal population. And we have a study ongoing in kids going from 6 months up to 12 years of age. So from the phase 3, something that also became clear is that if you look at the accumulation of cases in the placebo group, in the black line, and in the vaccinated group, you see that the 2 lines start diverging even before the second dose of the vaccine was provided, showing that the vaccine, at least again for the virus that was circulating at that time, was providing protection already after 1 dose, though the maximum protection and the long-term protection, it really requires the second dose of the vaccine. So, as I mentioned, there is, of course, we're looking at the longevity of protection, we're looking at the immunoresponses with time, and what we have seen after 6 months after vaccination is that the vaccine remains highly efficacious against the virus that was circulating, the Wuhan strain initially. So since then, and as Alessandro was mentioning, there have been a number of variants that have emerged in different parts of the world. And the main reason these variants are emerging is because the virus that has jumped from an animal species to the human host is actually adapting to the new host. So it's it's learning how to better replicate, how to better transmit from person to person, and to really, replicate better in the new host. And so that brings it to really a number of mutations that result in a virus that actually it's a better fit to the new host. But these mutants viruses, pose some risk to the human host. And there is a classification that has been, made by the WHO on the different variants. And, you will hear talking about variants of interest. So viruses that have mutations, so changes that potentially have an impact on or decrease in neutralization by monoclonal antibodies or immune responses elicited by vaccines. Variants of concerns that are variants where there is experimental actually evidence that these viruses have a higher transmissibility or that they cause more severe disease, so increase hospitalization and death, or decrease in neutralization of antibodies generated by immune responses elicited by vaccination. And then, there are variants that have not really yet been identified that there would be variants of high consequences, where there is a clear evidence of increased transmissibility and that these variants really don't respond to immune responses elicited by vaccination. So most of the variants that we are dealing with until now are either variants of interest or variants of concerns. And just to put this in perspective, I would like to, just to understand what these variants are, is mutations basically in different parts of the virus, but some that are of a higher concern are the mutations in the spike protein. So the spike protein that is depicted in this figure for different of these variants is the key that allows the virus to enter into the human cells and is the major target for neutralizing antibodies, monoclonal antibodies, or antibodies elicited by vaccination. And what you can see is that, depicted as a red dots are mutations or changes in the spike protein in the critical parts of the surface of the spike protein. And the variation are different for different variants. So here in this picture is depicted a variant that has emerged in the UK, the B117, a variant that has emerged in South Africa, the B1351, and variant that has emerged in Brazil, the P1, and the B1429 that has emerged in California. These are just some of the examples. And some of the mutations are shared among some of these variants, but some are different. So there are substantial differences in terms of escape to neutralizing responses among the different variants. And here I just would like to give you a sense of how these variants are, spreading in different parts of the world. Here is, for instance, a representation of the colors. It's a dark green will be the variant that has emerged in India, the Delta variant of 617.2. Light pink is the variant that has emerged in Brazil, and dark pink will be the C37, that is a variant that is dominant in for instance, in Colombia, in South America. But what you can see here, for instance, in very quickly since the end of 2020, in South Africa, there is a variant that is the P1 that has really taken over in Brazil, and this is really the dominant variant in Brazil. But if you then look in Argentina, you start seeing that there is not only the P1 but also this variant dark green that is emerging. And if you look in South Africa, where the South African variant has been really dominating since the end of 2020 for a large part of 2021, now is the Delta variant, so the variant that has emerged in India that has completely taken over. So one feature is that you can already see is that there is a changes really in the viruses that are dominating in different parts of the world, and they are not necessarily the same in different geographies of the world, that posing additional challenges for vaccine development. And so on. I mean, I have, of course, different examples, but here, for instance, you can see in Colombia, there is the variant was mentioned, the C37 that is taking over, but you don't have really the Indian variant at this stage. And to give you a sense again of the speed at which, this variant emerge and change, here is a representation of what's happening here in the US, where we have seen, first the emergence of the B117, so the variant that started in UK, but then since May is the Delta variant that is taking over, and at this point, actually I think it's more than 50% of the cases, though there is also some of the P1 variant that is still present in the US and actually has not disappeared with time. So the main challenge with this variant is that they, in some of these variants have a high transmissibility, as mentioned. So that means that they can spread very quickly, especially when there is a population that has not been vaccinated, with some variants taking over. And then some of the variants are really responding less well to the immunoresponses elicited by vaccination. So far, we know that the vaccines remain highly efficacious, especially after 2 doses against all the variants. But of course, there is always the risk that a new variant emerges that escape vaccine-elicited immunoresponses. And also, it's less well understood how the protection of the vaccine changes with time. So we know that the vaccine is highly protective, few months after vaccination. We don't know, in the long term, let's say 1 year after vaccination, what is the level of protection. And those are data that we are learning more from the real world, from, the use of the vaccine in different countries, for instance, Israel, where there's been a large vaccination campaign with the mRNA vaccines, and we learn more about the efficacy of the vaccine against different variants. And maybe I will finish by saying that this is how the Delta variants, so the variant that has started in India, is taking over in different parts of the world. But I'd like to mention that also, to be prepared for new variants, what we are doing as vaccine developers, we are also evaluating the how a third dose of the vaccine given, let's say, 6 months or a year after the primary series, so after the first 2 doses were given, how that changes the immunoresponses, and what is the best version of a vaccine that would need to be developed against an emerging variant. So those studies are clinical studies, also studies in animals that are currently ongoing, and it's are meant to be prepared in case a third dose would be needed against some of the emerging variants. So I'll stop there, and happy to answer to any question and look forward to the discussion.
Thank you, Andrea, and thank you, Alessandro. And of course, Andrea, last time we spoke was November, before the vaccine you helped developing was approved. I think we all owe you a big thanks for the work done since then. As scientists, we all hope to contribute to this world somehow. You actually accomplished that, so that's fantastic for us. So as we say, no question is a stupid question, and I see there's already a first question that's coming through the chat. Who wants to take that? Can a variant mutate and become more transmissible but less lethal?
Yeah, I think, maybe I'll start, and then so far, we have seen different variants. What the virus is doing, as I mentioned, is adapting to the new host. In the future, probably new variants will emerge that also will tend to escape to vaccine-induced or infection-induced immunoresponses. So the virus will evolve. Most of the variants that have really emerged and that are spreading are those that have higher transmissibility because they it provides an advantage to the virus and to in terms in evolutionary terms. We have not seen a decrease in pathogenicity in severity of disease. Perhaps in some cases increase, although it's very hard to demonstrate. I think there is a balance between the what the is in the interest of the virus in terms of evolution and the severity of disease, because of course the virus wants to have enough time to spread and to be transmitted. But I haven't seen cases yet of the virus becoming less pathogenic. However, what may happen with time is that as we get exposed to the virus or we get exposed to vaccination, is that the impact of infection is less severe, right? And so SARS-CoV-2 is not the first human coronavirus infecting the human population. I mean, we have seasonal coronaviruses that infect regularly the adult population and also children. And I think in that case, it's probably the virus has evolved for a long time with the human host. We have developed a sort of a defense or immune responses that control that infection, and therefore the severity of disease has become less, has decreased. So more than the virus, I think is the host that is getting, more prepared to fight the viral infection.
Well, I don't have to add anything to this very clear explanation. I just would like to point out that the virus really, it's constantly mutating, and there are lineages popping up here and there, and obviously in many cases we just don't talk about them because if they are not transmissible, perhaps they can can gain ground in some places and be spreading in some places, but they are in constant competition, and there are many other mechanisms why they might be successful or not. What we are looking at is the emergence of the more transmissible one that obviously do their job, find a way to transmit to more efficiently to the human host, and eventually also evading a little bit the immune system, so it's the virus is doing its job. It's I think the equation that the virus evolve always in a benign way. It's not something that is a given, and I think it's very nice what Andrea said. It's mostly the host that becomes more able to cope with the virus. So obviously, after many waves of infection, and then we have been primed with the vaccine, etc., our immune system, well, we will get more and more used to the virus. There will be less and less severe cases, and in a sense we will go into the classic endemic situation as we have for many viruses.
Thank you. We have a technical question for Andrea, and given the technicality of it, maybe it's better to be concise in the answer, otherwise most of the public will be lost. The question is, why do most variant mutations happen to the N-terminal domain of the virus? You're on mute, Andrea.
I think so. The most of the mutation emergence in different parts of the spike protein, but the one that allow the virus as a part of the improvement in fitness. So as the virus is adapting to the host, the parts that tend to change are the ones that are involved in the interaction with the human cells. And so the N-terminal domain and the region that is involved with the receptor binding are the most functionally important regions. And that's where the mutations are emerging, because the virus is changing the regions that are critical for interaction with the host. And so as it's trying to have a better interaction, that's the regions that it mutates. So the N-terminal region and the receptor binding domain are the regions that are critical for the interaction with the cells. They are also the critical target for neutralizing antibodies. So as we will develop immunity, I think those will be still the regions that will change, but for a different reason, probably to escape the immunoresponses rather than only to have to better fit the host. I hope
there's a natural selection.
Yeah, it's a different selection, different bottleneck. Yeah. We
have a next question which seems fit for Alessandro, which is it's an interesting question. So vaccines prevent severe cases and hospitalization. Great. But vaccinated people can still be infected. And so the question is, then how do vaccination decrease the development of mutations if people can still be infected? I don't know if it's Alessandro who's on mute now, or Andrea.
Well, yeah, well, this is, the vaccine works on many levels. The first one is that it decreases the probability of infection, so the susceptibility of the individual. It also decreases the transmission of that individual if it becomes infected with a more mild form of the disease. Then it protects against severe cases, and then eventually, obviously, against that. And so and I see in many cases that people think, okay, the vaccine is like something that is on and off. It's just 0 or 1. It's not like that. So the fact that we have a transmissibility reduction due to the vaccine means that we are already dampening the epidemic cures, and that we limit the transmissibility of the virus. It's not impossible that if you are vaccinated also with 2 doses, you can be infected. This is something that has been measured, and for the new variants is a little bit, this probability is a little larger than with the old variants. But you can imagine this is the transmissibility gets reduced, and so that the population is more protected. For the vaccine, we have to consider at this point that we had 2 major forms of protection from the virus. One is the natural infection, the other one is the vaccination. And at this point in the United States, as in many other countries, because of the vaccination and unfortunately the several waves of epidemic that have run through the population, we have a large fraction of the population that has been immunologically exposed to the virus. And that means that is, the fact that they get infected, or in some cases even they get reinfected, is very, very low. This is how we protect and slow down the virus. Unfortunately, the Delta variant. So a couple of months ago, we were almost in a situation in which we were just looking at a decline of the epidemic. Unfortunately, the Delta variant has a transmissibility that is so high that even the fact that there is a small fraction, unfortunately, 40% of the population is still not vaccinated, and probably less because of the natural infection. But this is a population that is still exposed, and where the epidemic runs at this point very quickly because of the high transmissibility of the Delta variant, and plus, the Delta variant has a certain probability of infecting even hosts which have been exposed to the virus. But I I won't put the things. What is the protection of the vaccine in terms of at the population level in terms of epidemiology? Is what the vaccine are giving us is a huge benefit in this fight.
And there was a paper published, I think about a month ago, looking at the Israeli population, who, as Andrea mentioned, was massively vaccinated, and showing how the transmission rates, once you have a vaccinated population, go significantly down. And even if a vaccinated person is infected, the amount of virus is minimal, which means it's harder, very hard for that person to further transmit the virus, which means you're reducing the steps and the passages of the virus, which the virus need to then generate variants, right?
Yeah, this is, I'm pretty sure that most of you have heard of this famous reproductive number, no? And what the vaccine does is essentially is to add a prefactor. So if you have that the reproductive number is 2, but the transmissibility is reduced by 50%, this number goes to 1. Unfortunately, now we are in a situation in which this reproductive number for the Delta variant is a number that is between 6 and around 6 or 7. Well, between, let's say, there is a lot of uncertainty between 5 and 7, and of course, even if you slash by a lot, this number tends to be larger than 1. There are other components, however. The number of individuals that you can infect is lower because of this exposure, etc., etc. So all that is creating this reduction of transmissibility that sooner or later should just bring us in a regime in which the disease decline until there is until some individuals becomes again susceptible to the disease. And here is an important point that Andrea, I think, was mentioning before. That is the waning of immunity that is unfortunately for some diseases happens on a certain time scale, from natural infection or from the vaccine. And so the need of, in that case, we have to think about the need of booster and other approaches to the mitigation and containment of the disease.
So we have a number of questions that came, and one follows what you were saying. Basically, what you're saying is really the more people get vaccinated, the more stops we have also for generation of variants, right? So one of the points that we wanted to discuss today is, why is it selfish interest for richer countries to support vaccination in third world countries? And as you explained before, third world countries are a pool of unvaccinated groups, large group of people who often live very closely to each other, and so where there's high transmissibility and therefore higher generation of variants. But you also mentioned how even in first world countries, 40% or so of the population is now vaccinated, and that still serves as a pool to generate more variants. So one question that came is, what can the World Health Organization or other organizations do to increase and support vaccination in countries where there's lack of supply, or in countries where there is supply but people are opting not to be vaccinated? So what else can be done to help India, Brazil, Africa to get the vaccine?
We are entering a question that is very complex. It's on many levels. It's not just epidemiological. This is a political question, it's an economic question, and then there is many phases to it. I would say the first thing is that for sure hoarding vaccine doses in specific countries that are not used, it's not wise. It doesn't help the country. They are just staying on a shelf, and doesn't help in how to say, instead of vaccinating people at risk in other countries in the first place, and then also other people, so that we can slow down even more the epidemic. So the first things is to think about donation for sure, and how to say, allow countries that are not able to acquire vaccine doses because of economic and resource constraint to get the doses that they would need. It's not just that, however, you know, we need also to, in many cases, to help the logistic effort in those countries. So it's you know, it's not enough just to send a certain number of doses on an airplane. But then we must be sure that those countries have an infrastructure that is able to put the vaccine in the arm of people. And in many countries, you know, there are all the possible complications that in the US or other European countries, we don't even think about it. That is the cold chain, how you reach out to remote villages in the middle of very hard to reach places. So it's really where also the WHO, but as well as other organizations, have to step up and help. You know, there is a lot of emphasis and has been put on the COVAX alliance that was building a stockpile of vaccine doses that were donated by different by countries, and that to be distributed. And so far, unfortunately, yet a small number of doses have been donated. There is one major thing, however, that we have to keep in mind, and I think that Andrea can probably speak much more informed on that. You know, at the bottom line, we need to produce more vaccine, and we need to have capacity, and that is something that has to be built from scratch. And we need to think perhaps not to this pandemic, but to the next one, how we can generate ways to have more supply. It's not just a matter of, at certain point, the bottom line is the number of doses available. And that's I think is the bottleneck, yeah.
Yeah, and the speed to make them available. Andrea, you work at Moderna, obviously. Are you aware of efforts that vaccine makers are doing to help distributing vaccine doses to third world countries?
Yeah, so I think in general, you know, for the distribution of vaccines is not something that at least Moderna is taking care of. I mean, during the pandemic, even in the US or Europe, this is done through, you know, local governments and so on and so forth. But there is efforts, of course, in terms of either donation or selling vaccines at low price to emerging, you know, economies. This is done through generally WHO, Gavi, CEPI. There's an organization called COVAX, and so both, for instance, Moderna and Pfizer have, you know, either donated or sold, again at the lowest price possible, the vaccine for, you know, use in those countries. I mean, at the end, I agree that there is there is a complexity, and in part is, of course, it remains, at least for 2021. I think we'll get better in 2022 about supply, you know, and a generation of you know, vaccine for all countries. The priority, of course, has gone to the, you know, countries that can buy the vaccines, but there is an effort, you know, from different companies, you know, AZ, J&J, and so on, to give vaccine to emerging countries. And also that has been done, it says, you know, the US itself, you know, they have started donating, you know, vaccine to other countries. Of course, you know, there are also concerns about the the potential use of a third dose. There's a prioritization that then happens. Yeah. But hopefully, for what Pfizer and Moderna have announced, and in terms of manufacturing for 2022, there should be doses for even emerging countries. I think the infrastructure for the use of those vaccines, it's an additional challenge. It's not an easy process to vaccinate millions of people.
So we have received, we have discussed a few very provocative questions for you, which we intend to ask because if it's not somebody present at the meeting, but I'm sure everyone knows skeptic friends who are not getting the vaccine. I'll just hold off one more minute before asking those couple of provocative questions. But one of those actually is, why should I be vaccinated when variants are emerging? And maybe there's no point in getting the vaccination. But you already both clearly explained how first, vaccines are slowing down the impact also of variants and are efficacious on preventing at least the more severe effects of an infection. And second, widespread vaccination does reduce the emergence of more variants. And variants have emerged in England, as in Brazil, as in India. They can emerge anywhere. Now, regarding variants, there's two questions which we can summarize as one, which is about are companies and developers working on new vaccines or variations of vaccines to protect against variants, and knowing the genetic code of the virus and knowing where most frequently these variants happens, are we able to predict what's the next variant emerging, and therefore already work ahead of time on a vaccine that will protect people for a future variant?
Yeah, so the first part, which is the simple one, I think all of vaccine companies are working on potential booster vaccines against the variants, with of course the challenges that are coming from the fact that these variants are changing, right? And so you have to assess what is the right vaccine that you want to develop. But I think that's work that is ongoing in all the vaccine companies. And I think Pfizer has, for instance, recently announced that they're developing a vaccine that is specific for the Delta variant, so the variant that has emerged in India, and Moderna has done studies using the variant that the sequence of the vaccine, if you wish, is the one of the virus that has emerged in South Africa. So it's of course in the, everyone is trying to be prepared if those vaccines were to be needed. Of course, at the end is up to CDC and regulatory agencies to both approve and recommend those vaccines if needed. And there is the other question is, predicting, what could be a vaccine that protects or covers future variants, or know if we can predict what is the new variants that are going to emerge. I think that remains extremely difficult. There are ways, of course, to do that experimentally and to try, for instance, to select mutants that are resistant to antibodies or to sera from people that have been vaccinated. But doing an ab initio, let's say, computational approach to predict what variant will emerge, I think that remains very hard, and even something that we have not been able to do, and remains a challenge, for instance, even for flu. I don't think anyone is able to predict, in silico, what sequence will emerge and what vaccine needs to be developed. Of course, there is a lot of efforts in this area, and I believe that in the future, in the next few years, we might get there with artificial intelligence, and maybe as we gather more information. But at this stage, I think it's extremely hard. I also want to point out that with the new technologies such as mRNA, bringing a vaccine to clinical study has become much faster. And so, within the 40 days, we can go from a sequence to a start of a phase 1 study. So let's say that the need to predict what variant is going to emerge, unless it's done with a super high confidence, it's not probably necessarily worth it. Yeah. But the short answer is that no, we're not yet capable of doing that. Yeah, unfortunately.
You mentioned the boost, and we read on the papers how the Pfizer has kind of leaked out that they're getting ready for a submission to the health authority for a third dose as a boost, and then Fauci confirmed that, and how the Pfizer CEO apologized to him. So there's a question about, first of all, do you think that we do need boosts? And I think there's a few papers, and you also suggested that earlier. And then also, would the boost dose, a third dose, be tailored to protect more against variants?
Yeah, I think that's the effort, right? I mean, it's is there a need for a third dose, so for a boost, and if so, what is the best vaccine to be used? Of course, as the vaccine is also used for for people that have not been vaccinated, probably you wanna, if possible, you wanna develop the vaccine that has the circulating sequence of the virus. That would be the optimal one. But then there are other questions that are related to the logistics, to the vaccine that governments might already have in their stockpile. So it becomes a little bit more complex. But in general, the effort is to try to develop vaccines that offer the best coverage against the circulating variants. And again, not that maybe of course I work on a mRNA technology company, but clearly the new technology allow to a certain extent to do that, in the sense that we can adapt very quickly the sequences of our vaccines and adjust to the circulating viruses. That is much harder to do with more classical technologies such as the recombinant proteins or replication defective or attenuated viruses.
So let's start with the first controversial question to interrupt the list of questions we have here. And let's
sorry,
start
I have to leave.
No, the one that is really circulating a lot, and is not just a myth, but a number of us have been told of friends or daughters of friends who are just getting married, and they are afraid to be vaccinated because they heard that getting a COVID vaccine can turn a woman sterile. This is a serious concern because many young women are afraid of that. So you tested Andrea, your company tested at least in the main phase 3 studies, then there's been additional studies in 15,000 people. Millions of people now have been vaccinated. Maybe what the public is not aware of is that once any drug goes on the market, any adverse event, any side effect has to be reported rapidly within 24, 48 hours by any physician to the health authority. Is there public, of course you cannot give confidential information, but has there any analysis been made on the rate of problems with pregnancy in women who have been vaccinated?
Right. I mean, I think there is two parts of the question. So first of all, we do studies not only before a vaccine moves to clinical studies. We also do studies in animal species, and we address some of those questions. Clearly, there's no from the data that we have, there is no impact using mRNA vaccines in general on sterility, reproduction, and so on. So there is a one part. So we don't see any impact. Vaccines, mRNA vaccines now have been given to hundreds of millions of people, men and women, and so we don't have any data that suggests that this is a problem. But also, the second question, that is a pregnant women, and so the vaccine has been given to lots of pregnant women. We have even data now on potential. There were questions about the presence of the vaccine in the breast milk. There's no evidence even for that. So pregnancy have been followed, and even immunoresponses, then that go pass from a woman to the child. So all those things are looked at, both in a real world studies. So people that have received the vaccine, but also in the clinical studies that are ongoing that have been designed to answer to those questions. So we have just started actually a clinical study in the maternal population to look at that. So it's important to remember that of course we do efficacy studies and clinical studies in thousands of people. And the Moderna study was 30,000. I think Pfizer was 45,000. But we don't stop there. Then we follow, the safety and efficacy of the vaccine in the population for years, and so that's the way we can learn about also rare side effects that are, cannot be identified during the clinical development of the vaccine.
Thank you. Other people are concerned that this mRNA, once injected in the body, will integrate into the person's body and DNA and be there forever or for a very long time. And therefore, they are concerned because of that, there could be long-term side effects that will happen only years later and therefore have not been seen in clinical trials. Now, maybe the one basis for the before your answer is also that many people have now worked in a lab with RNA, DNA, and mRNA, and so they don't know. And because we read that police was able to test DNA from a body which was left years and years and years ago. They think it's the same, but if you worked in a lab, well how while DNA is very stable, RNA is extremely delicate. So question, of course you don't have, question is, one, is there any biological mechanism for which an mRNA without a retrotranscriptase like HIV has can integrate itself? And two, have you done any measurement of how long does the mRNA injected in people, how long does it actually stay?
Yeah, I think it's of course there is a different answer to that question. I mean, but fundamentally the mRNA and the way it's designed cannot enter the nucleus of a cell, and so as such cannot integrate in the DNA. We also looked at that, of course, experimentally, and we have looked pretty hard actually at that question, and we have no evidences for RNA, mRNA being, getting inside the nucleus. And then also, those those observations that you were describing that are more in the context of a viral infection, require elements that are not present in the vaccine mRNA. And also the frequency and level of exposure, it's very low compared to what you have with the virus. In general, I can say that the mRNA, yes, is not very stable, and we can see that within 24 to up to 72 hours, it's fully degraded. So after that time, there's no more mRNA that at least we can detect after vaccination.
Okay, so to make it clear, you have run tests, and the mRNA is degraded within 24, maximum 72 hours.
Yeah. Yes.
Okay.
And it's not. We don't see mRNA getting inside the nucleus of cells.
For sure. Yes, because it would need a number of other proteins to be able to to do that. Thank you. Maybe we have another, of course, very controversial question, but maybe we'll switch to one question that is here in the chat, which is for Alessandro. Of course, it would be hard to vaccinate 100% of the people, and so the question is maybe not only how, but also, is anybody working on network data science to assess who's a high spreader, and at this point, once a good portion of the population is vaccinated, start vaccinating high spreaders rather than the more vulnerable who most likely have been vaccinated already?
Well, that's a classic point, and this disease, so SARS-CoV-19 has a profile in terms of risk that was so much skewed on elderly and related to age and the comorbidities that by looking at all possible strategies, it's the vaccinating first the population at risk was in terms of averted deaths the safest strategy. Now we are entering a different stage of the vaccination campaign, in which we are working at this point in, first of all, trying to convince people to be vaccinated. And so all the how to say controversial question that you are asking now, this is where we have to how to say to get the heart and mind of people in terms of vaccine are safe, and at this point the way out of this pandemic nightmare is the vaccine. On the other hand, we can think now of strategies that are more targeted to more active individuals, people who have large, how to say, more connectivity patterns that are more heterogeneous, more connects different groups, that are more for instance, that might travel more, etc., etc. So there are several approaches that have been developed through network science, that is the study of the connectivity pattern of people, the way we get together, we go to work, we commute, we travel, and that can guide, policies that optimize the vaccination campaign. It's not easy because there is a level of privacy that you don't want to infringe, and how to identify the I think in the question was the super spreader. It's very, very tough. Also, I don't want that there is some confusion about super spreading events that have been always put forward during this pandemic as responsible for large cluster of infection. They are not just related to the individual or to the biology of the individual or to the behavior of the individual. In some cases, it's also the setting, and so super spreading events happen because you have large congregation in small places with no ventilation, and this is something that is maybe it's just chance or happening, and it is not really related to individuals that we can target through vaccination. Although there are for sure, as in the question, perhaps retired people that do not see too many other individuals during a day, and perhaps an active for instance teacher or people working in large stores that can expose a lot of people to the disease. So yes, we can go and think about strategies in the future. At the moment, however, really for me the point is to convince every single person you talk to get vaccinated. And before, I think you said something about, what is the benefit of being vaccinated and the population, etc. Well, at the moment with the variant that we are facing, really probably the most risky activity is not get the vaccine. It's really it's so important to get vaccinated because it spreads so quickly, this new variant, that everybody who is not vaccinated is really at high risk.
Thank you, Alessandro. So then again, the point is to vaccinate as much as possible, produce as much vaccine as possible, and increase the distribution as much as possible. And to do that, we also need to keep solving the doubts of people who are not convinced yet, of course. So I really thank both of you for giving scientific answers to doubts that, if people are convinced of something, it means that they believe in some piece of data. And so it's important to explain with data and science what's the rationale for that doubt being not founded. One, maybe I'll get at one final, well, there's maybe a couple of more questions that came. One is, when do we know when a vaccine for children will be approved? I don't know if, of course, as you know, Andrea, we have many families in our community, and any update on when a vaccine may be approved, at least here in the US, for children?
Yeah, I think there is so going from 6 months to the 12 years of age, there are clinical studies that are ongoing, both Pfizer and Moderna. I think Pfizer is ahead. So I would expect there is a process that is age de-escalation. So you slowly go from the older children and go down with age. And while you do that, you test also the vaccine at different dose levels, because you want the vaccine not only to raise the immune responses, but also to be, well tolerated and safe in this population. So, it might be, it really depends on the discussions with the regulatory agencies and the path for approval. So if there is, an emergency use authorization or not. But I would expect that at least down to 5 years, probably sometimes in the fall, those vaccines could become available, and then down to 6 months towards the end of the year, or even beginning of 2022.
So 5 years and above in the fall, hopefully.
Hopefully, yes.
Yeah.
That's, I think, is the kind of timelines that.
One doubt that is also diffuse among parents is studies, while the safety of the vaccine is very well known for adults, the rumor is that clinical trials are testing a very small number of children, and therefore people don't trust the safety data from very, very small. Can you, it should be probably public information, it's probably on clinicaltrial.gov. What is the size of trials in children?
So if I'm not, it's a good question. If I'm not mistaken, the trial in the, in the pediatric population is around 6,500, and with a 2-to-1, so 2 vaccinated versus 1 placebo. Those are, of course, also discussions that are ongoing with the regulatory agencies, but that's normally the number of subjects that are exposed, and to look at even at the safety of the vaccine. So it's not the size of those studies is what normally is used for, let's say, approval or authorization of a vaccine in the pediatric population.
So because the side effect profile of drugs is not that dissimilar between adults and children, there's an extrapolation that you do from the safety from adults to children, and any other drug development for any other type of vaccine has similar numbers as the number of patients, of people, children being tested in this trial.
Yeah, I mean, I think there is, like a number. So, even even in the adult population, there is a number that is, is considered sufficient to, let's say, identify a safety signal. Then, for the rare events, still you have to go to very large, numbers, because you're not, even with the 30,000 subjects clinical study, you're not going to capture
Very rarely. Yeah.
So.
I mean, as a comparison in cancer, a phase 3 study, a big phase 3 study has 1,000 patients in total between treatment and control. So the study that you're running in children is 6 times bigger than a big study to approve an anti-cancer drug.
Also keep in mind, those studies are not easy. I mean, of course, you have to find people that want to be part of the study. They need to enroll, and there is a logistics. So there's a lot of complexity, and of course, the largest are the numbers, and the longest is going to take to bring the vaccine to the finish line. So.
The last controversial question, I promise, that I'll ask is something that also has circulated quite a bit, which is you, Andrea, you are head of research at Moderna, so obviously you oversaw the research to make this vaccine, and then you're involved in, of course, in the quality production and so on. If you wanted to trace people, geolocalize people by inserting something into a vial, which is kept at -80 or -20, and then at room temperature for a while, and then administered into the body of somebody. What type of, first of all, what type of device would that do that? And would the FDA, because when you get any drug approved, right, you have to demonstrate very clearly in an excruciating level of detail how you make that drug to the health authority, not one, but health authorities of every country. They want to know exactly the composition that's in every vial that you put into a patient. Do you think that if somebody really wanted to do it, there would be a way?
There's a few things I can say, but I'm sure this can be a long. So first of all, there is when you do a vaccination or for anything, there's an informed consent. So unless you are there's no regulatory agency or government that would allow you to put something in a vaccine that allow you to do any kind of tracing. So this would have to be something that we do without approval, would be a conspiracy by a company and someone else.
But the rumors are that the U.S. government is involved into
okay, no, that's fine.
people.
So we can keep going now, sure. So that would have to be an alliance, between the pharmaceutical and government. But going more at the technological level, I don't know how you could possibly do that. So, and the vaccine is stored a certain temperature, but is also administered through an intramuscular injection, has to go through a needle. It goes into, has to be tolerated. So I don't know, even conceptually how you can do that. And if you want to do that, probably I wouldn't do it when I give the injection, but rather when the person comes for the injection, I find a different way to do it. So the bottom line is that I cannot think of a way to do it. If someone can explain it to me, and then I would be interested in hearing it. But I think it's conceptually very hard or impossible for what I know.
And Andrea, I had bigger faith in your technological capabilities.
Yeah, but we're not working on the.
You need to do more research. No, but seriously, thank you. I mean, it's again, if something goes around, it means that there's enough number of people who think that could be possible. So to be able to discuss scientifically and based on data any type of question is very important. Otherwise, the skeptic will remain skeptic, and the people who are convinced will remain convinced. We need to try to bridge the gap. So I hope that the people.
But you could right, but it's there is always theory. So you can say, you know, if you drink tap water, you get a chip in your stomach. So we can say lots of things. Then you have to, you know, is it feasible? And then what are the data that support it? So.
And if the government wants to trace people, probably they just need to check your cell phone.
Exactly, they are doing it. I think.
Yeah, I think there was a rumor also that the 5G technology was somehow related to the vaccine, but unfortunately my phone doesn't work better than before I was vaccinated. So I think we have the proof that getting vaccinated has nothing to do with 5G telephone connections. I don't know if anybody has other questions. We went quite beyond the initial planned time. Obviously, that's because it was a very interesting and nonstop fireside chat with you. So again, thank you so much, both Andrea and Alessandro, for again dedicating time to us. Thanks to the volunteers who spent so much time planning the content, the design, and organizing this. Thanks to the Consul General for supporting us from the very beginning and for being connected so late from Italy. And thanks to all the organization in Philadelphia, Washington, D.C., San Francisco, Miami, and Boston who supported. Consul, did you want to say anything to conclude the event?
No, I think that it was extremely interesting, and you addressed many of the questions that I myself have heard from many people. So as I said earlier, I think that it's very important that the public out there gets the information from people who are qualified to give relevant, significant, and truthful information. So thank you, Giovanni, and thank you to the very, very talented speakers that we heard today. And thank you to the public for participating and for asking such interesting questions. Grazie, Giovanni.
And Andrea and Alessandro, last time we had also a few more participants. I can tell you we heard from many people feedback on how the information that was discussed last time, I'm sure this time as well, were then used by the participants to spread the information beyond the attendance to the meeting. As we said, we're recording this meeting. It's going to be on our website. Last time, hundreds of people watched it after, and I hope the same will happen now. I think everyone who can spread data and factual information will help getting further along the fight to this pandemic. So thank you again, everybody. Have a good night.
Thank you.
Thank you again.
Thank you very much, and have a good night.